We are all unique. Some of us are tall, some short, some large, and some are thin. Some of us are born to be engineers, while others are meant to be artists. But just what is it that makes us who we are? Inside each one of us are trillions of cells that make up who we are. Each cell is a whole system that is like a small factory, responsible for making proteins in your body. Proteins are the building blocks of life, and every cell in the human body contains protein. Your body produces or synthesizes millions of different proteins that are responsible for every task in your body, including protecting your body against bacteria and viruses, transporting nutrients, hormone production, making connections between neurons, all the way to creating your appearance. Perhaps the most important part of the cell is the nucleus. Inside the nucleus, there are 23 pairs of chromosomes for a total of 46. In a single pair, there is one chromosome from your mother and one from your father. Chromosomes contain the genetic code of your body in the form of deoxyribonucleic acid. This is the body's instruction manual that defines the sequence of amino acids by which proteins are synthesized. DNA is a very long, well-packed nucleotide sequence that is made of the nucleotides adenine, guanine, cytosine, and thymine. If you unwound and tied together the strands of DNA from just one cell, it would stretch almost 6 feet long, but only be 50 trillionths of an inch wide. If you stretched all of the DNA in your body end to end, it would reach to the sun and back over 600 times. There are several steps in the DNA replication process by which DNA makes a copy of itself during cell division. In order to synthesize proteins, DNA is transcribed into messenger RNA, which contains the coding sequence for protein synthesis. Ribosomal RNA molecules form the core of the cell's ribosomes, and transfer RNA molecules carry amino acids to the ribosomes during protein synthesis. Proteins are built from a set of only 20 amino acids. Ribosomes produce a chain of proteins until translation is terminated and the protein is completed and then folded. The flow of information from DNA to RNA to proteins is one of the most fundamental principles of molecular biology and is so important that it is sometimes called the central dogma. Just how important is DNA? The entire history of our planet, since the Big Bang, has been about the eternal struggle of all life, from bacteria to humans, to survive and propagate each species. So how did certain individuals adapt and survive different conditions, like the cold? Let's look at the amino acid sequence of a protein, where fitness can be understood as the efficiency of a protein performing its function. Horizontal lines show all kinds of amino acid sequences. The vertical lines show how well they perform their functions. Evolving through the accumulation of neutral or beneficial amino acid substitutions, a protein can move horizontally or vertically in this landscape. Descent into the valleys is prohibited because mutations that reduce the efficiency of the protein are eliminated by selection. By supporting beneficial mutations, selection attempts to drive the sequence as high up as possible. Let's look at a few examples of genetic evolution. American white-footed hamsters normally have a dark color. However, another species of this animal that lives in an area with very light soil has a lighter color than its relatives. This step of evolution helped the species become harder to see by birds of prey, and light-colored hamsters were more likely to survive in their surrounding habitat because of this. But what happened that changed their normal dark color to a lighter one? It turns out that in the light-colored hamsters, a mutation occurred that deleted 3 nucleotides encoding the amino acid serine from the sequence. Only 3 molecules, which aren't even visible under a microscope, allowed the dark-colored hamsters to change their fur to a light color, giving them a higher chance of survival from being eaten. Another example of evolution is rice. It is an ancestor of wild rice, whose seeds sometimes fell to the ground. These were not easy to collect, and a lot of rice was lost. But at one point, a mutation occurred in the seed-shattering gene, SH4, which was responsible for the formation of the release layer at the disconnection site. As a result of the mutation, the functionality of the seed-shattering gene in wild rice was disrupted, and the rice acquired the necessary properties to stop falling. One more example can be seen in dogs. Less than 100 generations ago, dog breeders bred a new breed of short-legged fox hunters. At some point during breeding, a mutation occurred on chromosome 3 in the FGFR3 gene. The protein encoded by this gene is called the fibroblast growth factor receptor. Mutations in this gene led to a variety of skeletal development disorders, including shortening of the limbs. Because humans wanted to select for shorter-limbed dogs, all dogs that had this mutation were bred, and puppies without it were rejected. All of these mutations show the importance of DNA. But could we make changes to our DNA to become better? Genetic engineering. Genome editing, which is also called gene editing, are technologies that give scientists the ability to change an organism's DNA. There are thousands of genetic disorders that can be passed on from one generation to the next, which are sometimes serious and debilitating. In fact, 1 in 25 children is born with some form of genetic disease. Gene editing has already been used to modify immune cells to fight cancer or develop resistance to HIV. It could also be used to fix defective genes in human embryos, preventing babies from inheriting serious diseases. Gene editing has also been used in the agricultural industry to create seedless tomatoes, gluten-free wheat, and mushrooms that don't turn brown. Drug companies working on next-generation antibiotics have developed otherwise harmless viruses that find and attack specific strains of bacteria that cause dangerous infections. One of the more incredible things happening is that researchers have begun to use gene editing to make pig organs safe to transplant into humans. The problem is that porcine endogenous retroviruses are permanently embedded in the pig genome, and research has shown that they can infect human cells, posing a potential hazard. Researchers in the US used the precision gene editing tool CRISPR-Cas9, combined with gene repair technology, to deactivate 100% of porcine endogenous retroviruses in a line of pig cells. Piglets cloned from the connective tissue cells turned out to be free of viruses. What is CRISPR-Cas9, and how does it work? The biggest breakthrough in gene editing, considered one of the field's greatest achievements, is a molecular tool called CRISPR-Cas9. It uses a guide molecule, or CRISPR bit, to find a specific region in an organism's genetic code, a mutated gene, for example, which is then cut by the enzyme Cas9. When the cell tries to fix the damage, it often ends up with bad results and effectively disables the gene. This can be used to disable and turn off harmful genes. However, other repairs are possible, and if a scientist wants to mend a faulty gene, they can cut the mutated DNA and replace it with a healthy strand which can be injected alongside CRISPR-Cas9 molecules. During some of the first gene editing trials, scientists collected cells from a patient's blood, made the needed genetic edits, and then injected the modified cells back into the patient. This gene editing approach looks promising as a treatment for people with HIV. When the HIV virus enters the body, it infects and kills cells of the immune system. In order to infect cells, and just like any other virus, HIV must first latch onto specific proteins of the surface of the immune cells. In order to combat this infection, researchers took immune cells from a patient's blood and used gene editing to cut out the DNA that the cells need to make the specific surface protein that the virus must latch onto. Without these proteins, the HIV virus can no longer gain entry into immune cells. A similar approach has been used to combat certain types of cancer by using the same method of taking immune cells from the patient's blood and using gene editing to produce surface proteins that bind to cancer cells and eliminate them. In 2015, a baby girl with aggressive leukemia became the first human in the world to be treated with designer immune cells that were genetically engineered to wipe out her cancer. doctors infused 1 ml of the genetically engineered immune cells under special therapy regulations in the UK. The cells came from frozen batches of donated T cells, a type of white blood cell which plays a central role in the human immune system. Before these specially genetically modified cells were infused, they were given an extra gene that would induce them to target and attack leukemia cells. They then disabled other genes to stop them from attacking the immune system of the recipients and to make them invisible to a drug called alemtuzumab, which is used as an immunosuppressant. Weeks passed, and the trial worked. The most impressive thing about modifying cells outside of the body is that they can be checked before they are injected back into a patient in order to make sure that the editing process has no problems. The creation of genetically engineered viruses. Engineered viruses are specially designed and modified. They include oncolytic viruses that are manufactured to target and destroy cancer cells while leaving normal cells unharmed. Oncolytic viruses are engineered to contain certain genes and with the ability to infect cancer cells and produce proteins that cause the cell to die. In animals, these viruses have achieved promising results in fighting a variety of cancers, including osteosarcoma, a form of bone cancer. So far, the problem in adapting this treatment to humans is that most people have antibodies to the viruses used in animal studies. Because of this, the immune system is able to destroy the viral agent before it has a chance to infect the cancer cells. In research trials, the adenovirus viral therapy has shown good results in fighting several types of cancers, including squamous cell cancers of the head and neck, and is being used for precancerous oral tissue. There is a new biological therapy that enhances the natural cancer-fighting ability of a patient's T cells, which are collected and mixed with viruses that carry a specific set of genes. These designer viruses infect and deliver the genes to the T cell's nuclei, where they become part of the cell's DNA. These genes then cause the T cells to generate a special protein, CAR, on the surface of the cell. Once these CAR T cells are infused into the patient's blood, they are now equipped with the ability to seek out, latch onto, and attack and destroy cancer cells. There have been some dramatic results using CAR T cell therapy in some children and adults suffering from leukemia or lymphoma, but there have been some severe side effects. Cancer vaccines are another form of immunotherapy. They involve gene therapy where tumor cells are taken from the patient and are re-engineered with genes that cause them to become more recognizable to the immune system. The altered cells are infused back into the patient with an immune-stimulating compound. Now, the boosted immune system can launch a powerful attack not only on the newly infused cancer cells, but also on other rogue cells in the body. We live in an exciting time, and this is just the beginning of the gene editing age. Who knows, maybe soon humanity could become immortal.